Everyone to the June 2023 Viruses and Vaccines Seminar Series. For those of you who don't know me, I'm Alpana Wagmare. And just a couple of announcements before we get started. So we won't be having a genomics update today, and actually we'll probably take a break from it for the summer as things have sort of slowed down. But if things change, we'll definitely add that back into the seminar series. So it's my pleasure to introduce Dr. Anne Durer. Dr. Durer is the Director for Scientific Affairs in the HIV Vaccine Trials Network, as well as Professor in the Vaccine Infectious Disease Division and the Public Health Sciences Division at the Fred Hutch, as well as at the University of Washington in the Epi and Global Health departments. So we're very excited to have Anne with us. She's been in the field of HIV research for over 2 decades. And I believe as a first for this seminar series, she's going to be talking to us about HIV vaccines. So looking forward to hearing from you, Anne. And I will stop sharing and let you share your slides.
Thanks, and it's a pleasure to be here. So thanks for the invitation. So just want to see now. Can you see my pointer?
Yes.
Yes.
Okay, great. Okay, thanks everyone, and good morning. I'm going to be taking you on a pretty fast historical tour through where we've been in HIV vaccine research for about the past 30 years. These are my disclosures. And I'd like to start with a summary for the community. So a few points. What's the main issue that I'm going to discuss in my talk? And it's really, I'm going to focus on trials that have tested the ability of HIV vaccine to prevent infection, the so-called efficacy trials. And I'll focus on first on where we've been, and then I'm going to spend a few minutes at the end to tell you about where we're going. And the key take-home message is that the approaches that we've used thus far in HIV vaccine efficacy trials did not result in protection against infection. However, recently there's been a proof of concept trial that showed that broadly neutralizing antibodies can protect against infection by HIV if it's sensitive to the broadly neutralizing antibody used. And why is this important? And because this recent result set off a pivot in the field towards strategies aimed at inducing and delivering broadly neutralizing antibodies to HIV. I want to just say that there is a very promising approach for T-cell based vaccine using CMV vectors, but I'm not going to cover that. That's been done by Louis Picker at OHSU, and I won't have time to cover that today. So where have we been now 40 years into the epidemic, and where are we today at this important point? We still believe that an HIV vaccine is our best hope for ending the HIV epidemic globally, and for several reasons. One is that HIV spreads very fast throughout the body shortly after HIV acquisition at a mucosal surface, and there really isn't time to intervene between the point of HIV acquisition and widespread infection throughout the body and seeding of the HIV reservoir. Another reason is that roughly half of the 1.4 million people who are infected every year do not have identifiable risk factors. So it's very difficult to get to these people and deliver them other prevention modalities such as PrEP. So really, to get this kind of global coverage, I would think that vaccines really are the only practical way to go about it. And so in this talk, I'm going to review for you where we've been, noting just the past efficacy trials. There's been many early phase trials that I won't have time to touch on. And then I'm going to say a little bit about where we're going. So this slide is just to remind you that in the vaccine effort, we want to harness both arms of the immune system. On the left, you see the humoral arm, antibody production. And what we hope to elicit here is neutralizing antibodies. Neutralizing antibodies are those that bind to HIV in a way that prevents it from entering its host cell, the CD4 cell. And in this way, neutralizing antibodies can prevent infection. In contrast, eliciting T cells, and here we're talking about CD8 T cells, can control or abort infection, but they can't prevent it. And this is because these cytotoxic cells destroy infected cells by recognizing a self and a foreign antigen on that cell, and then that cell is destroyed. So this is something that we see, for example, in elite controllers. We see high levels of these cytotoxic T cells directed against HIV-infected cells. And in the middle, you see CD4 positive helper cells that play an important role in the elicitation of both neutralizing antibody responses that prevent infection and T cell responses that can abort or control infection. So what are the targets for this kind of immunity? Shown on this cartoon of an HIV virion, you see that the only molecule that's expressed on the surface is HIV envelope. And this is comprised of GP120, a glycoprotein, and GP41, which tethers it to the membrane. These are glycoproteins that are presented as a trimer, and they don't actually look like this. This looks rather like a mushroom, but in reality, it looks more like a broccoli, that there's lots of indentations and crevices on the surface, which make it even harder to elicit antibody responses to this molecule. In addition, a number of internal proteins have been targeted by a variety of vaccines, proteins such as GAG, polymerase, NEF, and protease. And I'll talk about these a little bit more as I go through the individual vaccines. So now to move on to a history of the vaccine trials, I'm going to basically go through 3 phases. The first phase was using monomeric ONC-based vaccines. And this happened towards the end of the 1990s and in the early 2000s in the VaxGen trials. And the VaxGen trials used GP120 in an attempt to elicit neutralizing antibody responses. In these trials, 7 doses were given over a 36-month period. And there were 2 trials that were conducted. The first was conducted in subtype B regions in the US and Europe and enrolled roughly 5,000 MSM and a smaller number of women. In the second trial, conducted in Thailand among people who inject drugs, the vaccine used was a GP120 from not only a subtype B virus, but also the AE recombinant that was circulating in Thailand. These studies, unfortunately, did not result in protection against HIV infection. And you can see here on the right, a Kaplan-Meier curve starting with 100% of people without HIV infection. And as you can see over time, the study trial population accumulated HIV infections at exactly the same rate in the vaccine and placebo arm. So the antibodies induced by this HIV vaccine bound to HIV, but they did not prevent infection. That is, they did not neutralize. So these results led into the second phase of HIV vaccines, which took an entirely different approach. And it was realized that eliciting neutralizing antibody responses that could prevent HIV acquisition was a daunting task. And so the field turned to look at T-cell-based vaccines. And these were really grew out of the observation that CD8 responses seem to be effective in elite controllers in preventing them from, in controlling viral load, preventing CD4 decline, and many of the diseases associated with advanced HIV infection. So the idea was to try to make vaccines that would mimic it and prevent the reduction in CD4 counts and controlling viral load. And the hypothesis going into these trials was that the more potent the T cell responses, the better the vaccine. So in this phase, these vaccines were focused on eliciting T cell responses. And I'll go over 3 of them conducted between roughly 2004 and 2014. The first of these was the Step and Pombilli vaccine studies. And these used an adeno5 vector, so a common cold vector that had been attenuated and contained inserts to 3 proteins, GAG, POL, and NEF. And note that this vaccine did not contain any envelope. There was only internal proteins. And the idea was that this vaccine would result in elicitation of CD8 responses against HIV. And in fact, the Ad5 vector was very effective at eliciting CD8 responses. And using a prototype, we had seen control of viral load and prevention of T cell reductions in a non-human primate model. So there was reason to believe that this candidate could be effective in humans. So it was studied in 3,000 MSM in women in clade B regions, and as well as a smaller number of men and women in South Africa, hoping to elicit cross-clade protection. The endpoint from this study was really to prevent disease associated with HIV by controlling viral load and preventing CD4 decline. So it was anticipated that participants would acquire HIV on trial, but that they would be prevented from developing negative consequences from that HIV infection. So as you can see on this slide, there was no protection against HIV acquisition and no reduction in viral load in participants in these two studies. And in fact, in these results shown on the right from the STEP study, you can see now we're going in the opposite direction. This is cumulative number of new infections, that there were actually more new infections in vaccine recipients than there were in placebo recipients. And this was due to increased susceptibility to infection in subgroups of the study population, namely men who were uncircumcised or men who had prior evidence of immunity to Ad5. This increased susceptibility was transient and is still unclear what caused it. The next study was HVTN 505, which used a related but slightly different approach. It contained a DNA prime, shown here, 6 different DNA molecules as part of the prime, which contained clade A, B, and C envelope, as well as clade B, GAG, POL, and NEF. So envelope as well as the internal proteins. This prime was then boosted with an Ad5 vector, which was different from the Ad5 vector used in the STEP study. And this Ad5 vector contained clade A, B, and C envelope, as well as clade B GAG-POL fusion insert. The study was conducted in the US among Ad5 negative circumcised MSM. So it's important to note that there were several safety features here. First, the Ad5 vector that was used was not the same Ad5 vector used in STEP. And secondly, the study was conducted entirely in the group of participants who showed no increase in risk of HIV acquisition in the STEP study. This trial had 2 co-primary endpoints. The first to prevent HIV infection, and second to prevent disease, the second being the more important outcome of controlling viral load in participants who acquired HIV on trial. The results of this study showed no protection against HIV acquisition and no reduction in viral load. The next study then used a slightly different approach and a different vector. This was a very large study in 16,000 participants and used an ALVAC vector. So this is a canary pox vector, which is host range restricted in humans. And it was given as 4 doses, and the second 2 doses got a GP120 boost. And this boost actually was the AIDSvax boost, which I showed you several slides ago. This was the clade B and clade E or AE recombinant GP120 that had been used in the TIDE trial, the TIDE VaxGen trial. So this study was conducted jointly by the HIV research group at Walter Reed and the Thai military. As I said, it enrolled over 16,000 participants near Bangkok, roughly half men and half women at average risk. So the study did not seek out individuals at recognizably high risk of HIV acquisition. And it had 2 co-primary endpoints: to prevent HIV infection and to prevent disease by controlling viral load. And the results were somewhat different from what we've seen before. This vaccine regimen resulted in modest protection against HIV acquisition. And you can see that here on the right in the Kaplan-Meier, in the higher number of cumulative HIV infections in the placebo recipients compared to those who received the vaccine. It, however, had no effect on viral load, and this was not particularly anticipated from the kind of vaccine regimen it was. Importantly, the protection that we observed was correlated with non-neutralizing antibodies to the V1, V2 loop, as well as to CD4 responses, rather complex CD4 responses that gave a variety of different cytokine responses. So this result that non-neutralizing antibodies were associated with protection against HIV acquisition ushered in the third phase of the HIV vaccine in our history of HIV vaccine trials. And these were focused on non-neutralizing antibodies, and I'll review them here. The first was an attempt to repeat the success of the RV144 trial in Thailand by customizing the vaccine regimen for South Africa. And this regimen had several changes. It used the same Alvac prime, but this time now with a clade C envelope insert to match the clade in South Africa, as well as clade B GP120 and 2 internal proteins. It had a different GP120. It used a clade C GP120 and a different adjuvant, a more powerful adjuvant. In addition, it added 2 more boosts at 12 and 18 months in an attempt to increase the response, increase the magnitude of the response, as well as increasing the duration of the response. So the goal here was to test a vaccine that could be licensed in South Africa. And they considered this a first-generation HIV vaccine that could be useful for their country if it had a good safety profile and if it could reduce HIV acquisition by 50% or more. And the results of the trial are shown here. Importantly, there was no protection against HIV acquisition. As you can see, very disappointingly, in the Kaplan-Meier, the incidence in the placebo recipients and the vaccinees was virtually identical. And in fact, this trial was halted by the DSMB for non-efficacy. The second study in this phase was 2 studies, actually. HVTN 705 and 706 were conducted by the HVTN in collaboration with Janssen Pharmaceuticals. This was a study using a different vector, an adeno 26 vector, which contained a gag pole insert as well as envelope. And importantly, the inserts in this study were mosaic, and they had been generated in silico from fragments from different clades virus in an attempt to increase coverage. And the idea here was to try to generate a vaccine regimen that could be used globally for protection against multiple clades. The vaccine construct had shown protection from challenge in non-human primate models. 40 to 50% of animals were completely protected from heterologous challenge, and the protection was correlated with antibody levels and both binding antibody and ADCP, antibody-dependent cellular phagocytosis. So correlated with level and functionality of the antibody response. So this regimen, an AD26 prime with a GP40 boost, which was a GP40 boost was clade C for the clade C regions and clade C plus mosaic for the clade B regions. So this regimen was tested in 2 trials, HVTN 705, which enrolled women in sub-Saharan Africa, a clade C region, and HVTN 706, which enrolled MSM and transgender persons in the Americas and in Europe. And this had clade B plus mosaic as the boost. So mosaic prime plus a clade B and mosaic boost. And the results of these 2 trials are shown here. Unfortunately, there was no protection seen in against HIV acquisition in either trial. And you can see here the superimposition of the 2 curves in the Kaplan-Meier plot. So what now? I've just shown you a series of efficacy trials, all of which failed to provide protection against HIV acquisition. And up until this point, the focus had primarily been on non-neutralizing antibodies and T-cell responses. So it was pretty clear that these hadn't worked. And we turned then to really try to understand more deeply why it's been so hard to elicit protective responses. So here you can see a model of HIV envelope glycoprotein. And what you can see is that this molecule is a pretty complex molecule, and many of the complexities allow it to evade the immune response. You see in blue on the surface what's the so-called glycan shield, and these are carbohydrate moieties on the surface of the HIV envelope, which are the same as the carbohydrates in the rest of our body. So they're not really appropriate targets for protective immune responses. In addition, the conserved regions, which are shown here in gray, are buried within the envelope, and they're shielded by the glycan shield, shielded by these carbohydrate moieties. In addition, use of this as an immunogen results in many non-neutralizing antibody responses, which are essentially diversionary. So we are trying to see whether or not we can elicit protective neutralizing responses, which occur in a sea of non-neutralizing, non-protective responses. So it's a bit hard for us to follow and to predict what immunogens are going to uniquely elicit neutralizing responses. In addition, there are many variable, hypervariable regions shown here in red, which elicit very specific responses, but not responses that are going to be broadly neutralizing, not responses that are going to protect at a population level. And then finally, HIV globally is hugely diverse. The reverse transcriptase of HIV is very error prone, and therefore many mutations are introduced during a normal replication cycle. And as a result, the diversity within a person, in the HIV infected person, the diversity in HIV is roughly the equivalent to the diversity in influenza globally. So it's very hard to elicit responses that can cover this level of diversity. The next reason why it's so hard is because it's very hard to elicit high level or high affinity and high avidity responses. And in these models shown here from cryo-EM images, you see in white HIV glycoproteins, the envelope on the surface of an HIV virion. And what you can see here, what you can appreciate is HIV is, if you will, bald. There are very few envelope proteins on the surface. They're few and far between. And this really poses a problem for antibody responses because it's very difficult for a single antibody molecule to cross-link between two envelope spikes. And if this could be achieved, we could get much higher avidity binding than we do at present. And we really need high avidity and high affinity binding to prevent acquisition. And one thing that we should bear in mind is for some diseases, we are able to prevent disease or prevent symptomatic disease without achieving sterilizing immunity. For example, for SARS-CoV-2 infection, we could protect against symptomatic COVID without protecting against infection with SARS-CoV-2 completely. That's not true for HIV. We need, as far as we know, we need sterilizing immunity. Most infections are established by a single virion. So if we can't prevent that single virion from crossing a mucosal surface, we can't prevent HIV infections. So the bar is really high. So we need, as I said, very high affinity and very high avidity responses. So how can we overcome these obstacles? And really what we've been focusing on lately is trying to elicit broadly neutralizing antibody responses. And these are responses that bind to very conserved elements on the envelope protein. The antigen binding regions are very specific. They undergo a lot of somatic hypermutation, and they can bind to these bind to these conserved regions and neutralize multiple strains. And we've seen in animal models that if these can be produced, they can provide sterilizing immunity in non-human primate models. So recently, we've been able to isolate and characterize many more broadly neutralizing antibodies, and some of them can now be made in large quantities in vitro in mammalian tissue culture. And as a prototype, these occur in many viral infections. Broadly neutralizing antibodies are induced and they provide lifelong immunity. And so if we have some hope that if we could induce them by a vaccination for HIV, we might be able to elicit long-term immunity to HIV. And this next slide shows you a few such molecules. This is the CD4 binding site, VRCO1. Next, the V1, V2 site for broadly neutralizing antibodies, the V3 site, and then the membrane proximal external region binds here to GP41. And you can see that it's quite tight there. Again, the CD4 binding site, VRCO1, I'll tell you more about this later, the V1, V2 binding site, the V3 binding site, and then the membrane proximal external region, which is a very tight fit there on GP120, excuse me, GP41. So we took advantage of the fact that these broadly neutralizing antibodies could be produced in mammalian tissue culture to do a passive immunization study. And in these, it was actually two studies, HV10 703 and 704. And in these studies, antibodies that had been produced in vitro were infused into study participants at 2-month intervals for a total of 18 months. And this study used, as a total, total of 18 months. So these used VRCO1, which is a CD4 binding site, broadly neutralizing antibody, and was conducted in 2 cohorts on 3 continents. HV10 703 was conducted in women in sub-Saharan Africa, and HV10 704 was conducted in MSM and transgender women in North and South America. So more than 3,000 people got VRCO1, broadly neutralizing antibody, infused passively in 2 doses with 1,500 control participants. And the objective of this study was to assess the safety and tolerability of this approach and to test its efficacy to prevent HIV infection. And you can see here the results. Protection was seen only against very sensitive HIV strains. There was no overall protection. So if the sensitivity to VRCO1 was high, as you can see, there was a quite high prevention efficacy of this approach, 75%. However, these strains of HIV that are highly sensitive to VRC turned out to be fairly rare. So there was no overall protection. And those that had intermediate or low sensitivity to VRCO1, there was no protection at all. So since these are the predominant strains, the overall efficacy was not significant. So what now? The next step then was really to focus on these promising results. We had proof of principle that broadly neutralizing antibodies, that is epitope-specific antibodies, could prevent HIV acquisition by strains that were sensitive to that broadly neutralizing antibody. So the 2 approaches that are being investigated now are first to improve the potency and half-life of broadly neutralizing antibodies, to improve the efficacy of passive immunization strategies. So basically to improve upon the results that I just showed you from HV10 703 and 704. And the next was to explore new and admittedly more complex strategies for inducing broadly neutralizing antibodies through vaccination. And I just want to note that, as I said at the beginning, there's also a T-cell approach being pursued by Louis Picker at OHSU, which looks very promising using a CMV vector, which I won't have time to discuss here. So back to broadly neutralizing antibodies then, the first step was to try to increase the half-life. And because as you noted in the previous trials, these broadly neutralizing antibodies were infused every 2 months, which is both expensive and not all that convenient for study personnel and for participants. So a mutation was introduced into the FC region of VRCO1, which decreases its clearance by increasing recirculation. And as you can see, after this mutation was introduced, the half-life of VRCO1 was increased about 4-fold from roughly from 2 months to roughly 8 months And so these modified monoclonals are what's now being used for trials being designed, early phase trials being designed now. The next step was to try to increase the potency. And here you see on the left a diagram of the potency of VRC01, the parent molecule, against placebo viruses that were isolated from the placebo arm of HVTN 703 and 704, the so-called antibody-mediated protection studies. And in this diagram, you see the viruses right around the periphery here. And these rays, different color rays, show the concentration of VRC01 needed to achieve 80% neutralization of individual viruses. So in this diagram, shorter is better. You need lower concentrations here. You need lower concentrations here to achieve neutralization of these viruses than you do, for example, for the green or purple or pink rays here, where high levels of VRC01 are needed to neutralize against these viruses. So in order to increase potency, a variety of mutations have been introduced, and I'm showing you here one of these, VRC01 23LS. And here you can see in the same format, you can see that the rays here are much, by and large, shorter, indicating that lower concentrations of this modified VRC01 are needed to achieve 80% neutralization of the viruses shown here around the periphery. The next step was then combining monoclonals. And this is just a schematic of some possible ways to combine different monoclonals to achieve broader protection. And we knew from our previous trials with VRC01 that a single monoclonal was unlikely to be sufficient. So if you can just imagine here an array on the y-axis, an array of different virions, you can see on the left what you might achieve from combining the membrane proximal external region monoclonal with a CD4 binding site monoclonal. And you can see that this is very broad. You have both monoclonals recognize all the strains in this panel. However, this membrane proximal region monoclonal antibody is not very broad. So it's not very potent. So this combination would be broad, but would not be highly potent. In contrast, on the right, you see a V2 apex and a V3 glycan monoclonal shown together. And what you can see, both of these are very potent, but they're not as broad. And in fact, most of the virions are recognized by only a single monoclonal. So this combination would be potent, but would not be broad. And then you see, what we would consider a more optimal combination of 3 monoclonals in the middle, where each virion is recognized by at least 2 broadly neutralizing antibodies that are both broad and in this case, potent. So we would have at least 2 potent broadly neutralizing antibodies for each viral strain. And it's regimens like this that we hope could be put together for passive immunization strategies in the future. But the next step would be moving to active immunization. How can we move from these passive immunization strategies to what we normally think of when we think of vaccines, active immunization or active elicitation of broadly neutralizing responses? And why has this been so hard? And it's been hard for a number of reasons. The first is the characterization of broadly neutralizing, or the characteristics of broadly neutralizing antibodies themselves. They have unusual structures with particularly long antibody combining regions, the so-called HCDR3 regions. And these unusual structures are a product of lengthy development, lengthy periods of somatic hypermutation in germinal centers. In addition, this process of evolution towards broadly neutralizing antibody is disfavored for a variety of reasons. The first, very importantly, is that the germline precursor does not recognize the broadly neutralizing antibody antigen. And what I mean by that is if you took a broadly neutralizing antibody and constructed its cognate antigen, the antigen that fits into that broadly neutralizing antibody in this lock and key configuration, if you took that antigen and showed it to the germline precursors, they wouldn't recognize it. So you can't start with the thing that you hope to end up with. So this really is a multi-step process. So since the precursor is stimulated with something that does not look like it could elicit a broadly neutralizing antibody, you need lots of somatic hypermutation along the way to get to a broadly neutralizing antibody. And the mutations leading in the direction of broadly neutralizing antibodies are rare, and some of the steps along the path are disfavored. For example, some produce autoreactive antibodies. So it's really a long and difficult path to get from stimulation of the germline to production of the final broadly neutralizing antibody. And then finally, we need response to more than one epitope. We learned from the studies that were conducted with VRC01 that we need durable, high level or high titer, and polyclonal responses in order to get protection from HIV acquisition. So what would be required to produce these protective antibodies? The first step here is stimulating the broadly neutralizing antibody precursor B cell. These are rare, they're diverse, and they're difficult to activate with HIV proteins. So then we need to get from this first step of stimulating the naive B cells to the final broadly neutralizing antibodies. So the strategy here would be to start by stimulating with something that can stimulate this precursor B cell, which probably does not look a lot like an HIV envelope. And then to guide the resultant B cells, to guide them towards plasma cells that produce broadly neutralizing antibodies. And this probably will need to be done in a series of steps. The first step would be shepherding the response to make it a little bit more like a broadly neutralizing antibody, using antigens that resemble HIV envelope a little more than the one that was used to stimulate the germline. This shepherding step might have to happen several times before we had the final polishing step, using something that looks like HIV envelope to produce plasma cells that produce broadly neutralizing antibodies. And throughout this process, there would be a large amount of somatic hypermutation that would have to occur to get from the germline to the final broadly neutralizing antibody. And this would have to be done a number of times in order to get different broadly neutralizing antibodies that could be act in combination to prevent HIV acquisition. So the idea is that the immunogens would set off maturation pathways that would ultimately lead to CD4 binding site, broadly neutralizing antibodies, V2 glycan, V3 glycan, and perhaps one of these, either the membrane proximal region or the fusion peptide. Some of these need germline priming. However, some of them do not. So perhaps it may be, if we only needed 3 of these, we might not have to use all of these that require this very long, arduous maturation process. So the VTN is investigating these approaches in something called the Discovery Medicine Program. And these are early phase trials that are focused on eliciting epitope-specific broadly neutralizing antibodies. And the idea here is that it's a rapid iterative process. These are small trials with a limited number of participants that use templated protocols. So the process of developing the protocol and implementing the trial can be quick. And the goal is really to test approaches for germline targeting and for maturation of broadly neutralizing antibody responses using different antigens and different adjuvants. And after these antibody regimens have, these vaccine regimens have been delivered, then there's a detailed laboratory analysis of the magnitude and specificity of the antibody responses. once these have been analyzed, they feed back in a feedback loop to the next trial, which would use modified different antigens, perhaps different adjuvants, to try to more effectively guide this process from the germline stimulation to the production of broadly neutralizing antibodies. So who's involved in this? Most of the trials I've told you about come from the HIV Vaccine Trials Network, which is a global network of scientists, researchers, doctors, community members, committed to ending HIV through the production of an effective HIV vaccine. This picture here shows you the trial sites on 4 continents. It's the largest clinical trials platform of its kind. On the right is something more recent and local. This is the Red Ribbon Registry, which was created recently, modeled on some efforts that investigators at the Hutch put into place during the COVID vaccine trials. And basically, this is a website which displays local clinical sites conducting HIV vaccine trials in an effort to try to reach communities that have been affected by HIV, but are less likely to participate in HIV vaccine trials, namely certain racial ethnic groups or certain social groups who have not routinely participated. And this approach was quite effective in increasing outreach for COVID vaccines. And so we've put it in place recently in an attempt to increase our outreach for HIV vaccine trials. So in conclusion, despite other prevention methods, an HIV vaccine, I hope I've convinced you that an HIV vaccine remains our best hope to eliminate the HIV pandemic, and that hopefully it will soon be within our grasp. The trials that I outlined for you, these landmark studies, all failed to show vaccine efficacy. However, recently, there's been a proof of principle study which led us to believe that protection by broadly neutralizing antibodies should be possible in humans. And we're definitely pursuing that, as I mentioned, in this discovery medicine approach that we're using in iterative smaller trials to try to come up with regimens that could be tested in efficacy trials, hopefully within the next 4 to 5 years. Finally, I'd like to acknowledge the study volunteers, the sites, the protocol teams, really for their dedication and commitment. This has taken thousands and thousands of people to get this far. And really, it's a tribute to all of them that we know as much as we do. And I'd like to acknowledge my colleagues and friends that have shared their slides and their work in progress. And I'd just like to leave you with this slide showing the incredible range and the amount of time it takes to get an effective vaccine. As you all know, this on the bottom of the slide, the SARS-CoV-2 vaccines were really a home run. They were really very quickly formulated and launched in trials, and amazingly, they were very highly effective. It hasn't been the case for many other vaccines, and here we sit at 40 years and counting, somewhere in the middle of the pack. So hopefully soon we'll be able to take this out of red and take this question mark away, and we'll have an effective vaccine. But I think it'll be at least another 5 years before we have results from the next set of efficacy trials. And with that, I'd like to thank you for your attention and take any questions.
Great, thank you so much. And what a great summary of I guess the last 40 years or so of research in this area. I wanted to welcome questions from the audience. So either in the chat or definitely feel free to unmute. I have a smaller group today, so ask your questions. I guess maybe I'll start with one, and apologies, I hope I didn't miss it, but where do you see mRNA vaccines, and I guess what's the status right now for that approach?
So thank you. I mean, it's obviously very promising, and we're looking into it, it would be a way, to deliver. It's not, we need first to figure out what the immunogens are going to be, and then, we can test the ways that we can deliver them. There are trials planned using mRNA. So I think, and we think, obviously there's a lot of experience now. It should be possible. But I think one of the issues for us is that we don't yet have the immunogens.
I have one more question, I guess, while we're waiting for others to think if they have anything. But maybe you did such a great job with the review with the last, not that many questions. In terms of the passive immunization strategy with the AMP trials, I guess I wasn't clear if that would be potentially an actual path forward for prevention, or is it more a means to sort of test out the efficacy of different antibodies?
Depends on who you ask, I think.
Okay.
So, there are many small molecule PrEP proponents who think that we're kind of there. We have TDF-FTC, we have long-acting cabotegravir, maybe we're going to have lenacapavir in the not too distant future. So, I think that especially, so there are a number of people that think that why do we need something else that would be, another type of PrEP. I think that both HVTN and IAVI are pursuing this passive immunization strategy with the idea that this could be potentially once every 6 months, maybe even once every year infusion, potentially also given subcutaneously. That could be protective. There's also a lot of interest in sub-Saharan Africa about using this approach in HIV infected women who are to protect infants who are breastfeeding. So basically the idea would be to give the breastfeeding infant who's HIV negative but exposed broadly neutralizing antibodies that could remain for 6 to 12 months. And, maybe be boosted once or get for whatever the duration of breastfeeding is. And the idea is that this could be long acting, maybe without some of the complications that you might get from giving a newborn a small molecule PrEP. So there's a lot of interest in this for that application. But it could also be, and also for infants, they're small. They need relatively small amounts of broadly neutralizing antibodies. It could be quite cheap. It could be delivered subcutaneously. So be, relatively easy to administer instead of using infusions. So, so I think that there are applications where I think it might be used. And there's also some discussion about cost. Could this be cost effective as well?
Okay, I have one question. Yes, it was directly to me. Has recent vaccine hesitancy had any effect on perception and involvement in these studies?
Wow. Yeah, I think it's a slightly different kind of vaccine hesitancy. Because treatment is really excellent. There is some complacency about HIV vaccines, and so I think it also depends on where you are. In the U.S. versus sub-Saharan Africa, and I think in sub-Saharan Africa there really is still a lot of urgency because of some of the difficulties in ensuring uninterrupted access to antiretroviral therapy, the desire, the stigma, the desire not to be on ART. So I think there really is, and the huge economic costs. So I think there's really a lot of urgency around HIV vaccine efficacy trials, and many of those are conducted outside the U.S., frankly, among MSM in Latin America and among women in sub-Saharan Africa, where really huge rates of HIV acquisition still continue. So I don't know if that answered the question. It's not exactly the same as the HIV, the vaccine hesitancy that we see for COVID, but it's definitely, there's definitely complacency that makes it a little harder to recruit.
Yeah. Okay, I guess two questions from Lauren. Are these vaccines being designed to elicit tissue-resident B cells at key mucosal tissues as a way to block the virus prior to reaching blood in specific types of infection?
So I think so. Are they specifically being designed? You know, it's not. I don't know if we're quite there yet, but what we do know from the passive immunization study actually is that there was quite a bit of accumulation of antibody at mucosal sites, and the half-life at mucosal sites appeared to be longer than it was in the periphery. So that's hopeful. You know, there were no B cells involved in those experiments. That was passive immunization of preformed antibody, but there isn't any reason to assume that if you're making the antibody, you know, the same antibody, that it wouldn't accumulate at mucosal surfaces. So that was, I think, quite promising.
Another follow-up question or separate question, will the diversity of BCRs elicited by the stepwise vaccines be measured versus only high affinity for the targeted strains?
So this is presumably a question for the, we're not there yet. So this is a question. We do have the first step. So there is a molecule that's been generated by sort of paring down envelope, which will stimulate the precursor. But in terms of, you know, I think that we probably will make some effort to measure the diversity of the response. But, you know, there's broadly neutralizing antibodies in natural infection occur at about not really before 4 years. So there's a huge long period of somatic hypermutation before you get to a broadly neutralizing antibody. So I'm not sure that we would, you know, try to follow every single, because there are a lot of blind alleys, right? There are a lot of things that don't go forward, especially like the autoreactive antibodies. So I'm not exactly sure what the question was, but we are, you know, trying to, I mean, certainly the focus will be trying to get to that molecule that's broadly neutralizing, a molecule that's broadly neutralizing. And there are some, you know, there are some steps along the way, you know, is this, do we reach what we think would be a lineage intermediate, you know, along the way. And there have been efforts to follow individuals over time. So really to have stored samples and then go backwards. So find an individual who made a broadly neutralizing antibody and then through stored samples go back and reconstruct the lineage.
One more question. If germline antibodies don't recognize HIV, what gets the response going in people who are naturally infected?
Yeah, so you know, so we don't, you know, I don't think I can necessarily answer that. But we do know from this molecule that was produced, it's a pared down piece of envelope. So, you know, there could be, you know, some partially degraded molecules, you know, some molecules that result from deletions or stop codons that still get produced that can bind the precursor. So it's not really clear. The molecule that was produced, that was used in vaccine, in a vaccine quote study, was pared down envelope that was then made into a polymeric structure so that it could have the advantage that it was polymeric instead of a single molecule. Whether or not that actually happens in vivo, I don't know.
All right, last chance for questions. All right, if we don't have anybody else, thank you so much for a great talk. So nice to get this overview and exciting changes in the field. So thank you very much. I did want to remind everybody about our July meeting, sorry, with Dr. Sean Murphy from Seattle Children's, who's going to be talking to us about malaria vaccines, and that's on July 18th, at the same time, 9 to 10 a.m. Thank you, everyone.
Thanks, everyone.